The Teloschistaceae are a large family of mostly lichen-forming fungi belonging to the class Lecanorrhomycetes in the division Ascomycota. The family has a cosmopolitan distribution, although members occur predominantly in temperate regions. Most members are lichens that either live on rock or on bark, but about 40 species are lichenicolous, meaning they are non-lichenous fungi that live on other lichens. Many members of the Teloschistaceae are readily identifiable by their vibrant orange to yellow hue, a result of their frequent anthraquinone content. The presence of these anthraquinone pigments, which confer protection from ultraviolet light, enable this group to expand from shaded forest habitats to harsher environmental conditions of sunny and arid ecosystems during the late Cretaceous. Teloschists flavicans is the type species of the type genus of the family Teloschistaceae. Regarding Teloschistaceae, and regarding scientific classification, its domain is Eukaryotae. Its kingdom is fungi. Its division is Ascomycota. Its class is Lecanorrhomycetes. Its order is Teloschistaceae. Its family is Teloschistaceae. 1898. Regarding type genus, Teloschists Norman, 1853. Regarding subfamilies, Caloplacoideae, 37 genera, Teloschistoideae, 33 genera, and Xanthorioideae, 45 genera. Regarding synonyms, Caloplacaceae. 1907. Teloschistaceae lichens typically have one of a few physical growth forms. Depending on the species, the thallus, the main body of the lichen, is either leaf-like, foliose, bushy, or shrub-like, fruticose, or crust-like, crustose. These lichens typically partner with a photosynthetic companion, a photobiont, from the green algal genus Treboxia. Teloschistaceae members are also characterized by their apothecia, the fruiting bodies where sexual reproduction occurs, which generally have a well-defined encircling rim of tissue. In the Teloschistaceae, the tip of the ascus, the structure that produces spores, characteristically turns blue when stained with iodine. The ascospores are released through a longitudinal slit in the ascus tip, a unique trait common to this family of lichens. The family, first formally proposed in 1898, was extensively revised in 2013, including the creation or resurrection of 31 genera. 3 subfamilies, Caloplacoideae, Teloschistoideae, and Xanthorioideae, are recognized. Since 2013, several dozen new genera have been added to the family, but there has been some debate about these additions. Ongoing DNA studies are helping to provide clearer insights into how the different groups within this family are related. The family contains more than 800 species in around 120 genera. 3 species from the Teloschistaceae have been globally assessed for conservation status, and others, such as the rare New Zealand species Caloplaca alanii, appear on regional lists. The full diversity of this family remains underexplored in vast regions like South America and China. Regarding human interactions and applications, although lacking any major economic impact, several rock-dwelling Teloschistaceae species are known to damage marble surfaces, and others are used in some traditional medicines. One member, Russifskia elegans, is used in research as a model organism to investigate resilience against the harsh conditions of outer space. The common and widespread Teloschistaceae species Xanthoria parietina, left, and Teloschists chrysophthalmus, right, were 2 of the earliest lichens to be formally described. The first members of the present-day Teloschistaceae to be formally described were the common sunburst lichen, Xanthoria parietina, and the gold-eye lichen, Teloschists chrysophthalmus. These were 2 of several dozen lichen species described by the Swedish taxonomist Carl Linnaeus, the former in his influential 1753 treatise Species Plantarum, and the latter in his 1771 work Mantissa Plantarum Altera. In his 1852 work Synopsis Lichenum Blasneosporum, Synopsis of Lichen Blasneospores, the lichenologist Abramo Bartolomeo Massalongo attempted to classify what he called Blasneospore lichens. This term referenced species, diverse in growth forms and appearance, united by the distinct polarilocular spores now attributed to the family Teloschistaceae. These are spores that are divided into two compartments, locules, separated by a central septum with a small hole. Although Massalongo's efforts to arrange these taxa into more natural genera were largely ignored by subsequent researchers, several of his proposed genera were resurrected for use 16 decades later, such as Blasnia, Gyalelechia, Pyrenotsmia, and Xanthocarpia. An ascus of the cruise-toes lichen Pyrenotsmia variabilis, containing 8 polarilocular ascospores. The family Teloschistaceae was formally circumscribed by the lichenologist Alexander Zylbruckner in 1898. In his initial version, he grouped together foliose and fruticose taxa having polarilocular, i.e. 2-locule, or 4-locule ascospores, including the genera Xanthoria, Teloschists, and Lethariocies. At that time, the growth form of the lichen thalia was often used in classical lichen taxonomy to segregate groups of species into families, and so in a subsequent, 1926, publication, Zylbruckner introduced the family Coloplacaceae to contain cruise-toes lichens with polarilocular ascospores. This family included the genera Coloplaca, Blasnia, Bombyliaspera, and Protoblasnia. The distinctness of the family Coloplacaceae was largely rejected by other authors, and it is now a historical synonym of Teloschistaceae. In another older classification, cruise-toes genera were grouped together in the family Blasniaceae or the Placodiaceae. In 1971, Carol William Dodge proposed the family Xanthoriaceae to contain Xanthodactylon, Xanthopeltis, and Xanthoria, but it was not validly published. In the 20th century, particularly with the widespread use of electron microscopy, the details of ascus structure became important considerations in the taxonomy of lichen-forming fungi. Studies on several Teloschistaceae species have noted the consistent presence of a cap-like zone at the tip of the ascus that shows a strong reaction to iodine, characteristic of amyloid substances. Using advanced transmission electron microscopy, Rosemary Honegger confirmed a unique type of ascus in Teloschistaceae, later named the Teloschists type. This ascus is distinguished by a special outer layer that reacts to certain stains and lacks the typical structures seen at the tip, opening in an unusual pattern during spore release. The presence of this ascus type was later used as a diagnostic character for the family Teloschistaceae following an ultrastructural study that corroborated Honegger's work. In 1989 Ingvar Carnefelt revised the family, accepting 10 genera, and this served as the main taxonomic classification for the family until the molecular era. In one of the last classifications of the family before the widespread use and implementation of molecular techniques, the outline of the Ascomycota accepted 12 genera in Teloschistaceae in 2006, Coloplaca, Cephalophysis, Fulgensia, Ioplaca, Josephpolcia, Cirriphora, Teloschists, Xanthodactylon, Xanthomendoza, Xanthopeltis, and Xanthoria. The family continues to undergo significant changes. For example, in 2020, of all fungal families, Teloschistaceae had the fourth highest number of new fungal names, a total of 128, including 8 genera, 48 new species and infraspecific taxa, and 72 new combinations. As is standard practice in botanical nomenclature, the name Teloschistaceae is based on the name of the type genus, Teloschists, with the ending ace indicating the rank of family. The genus name, assigned by the Norwegian botanist Johannes M. Norman in 1852, comprises 2 Greek words, tau lambda omicron, telos, meaning end, final, or term, and sigma chi iota sigma tau, schistos, meaning divided into, split, or separated. It refers to the split ends of the thallus branches that are characteristic of that genus. Cladogram showing the phylogeny of some species and genera in family Teloschistaceae and in the order Teloschistaceae, based on analysis by Wilk et al. In 2021, simplified from original. Species names have been updated to reflect current taxonomy. Single quote marks around a genus name means that the species is likely misclassified. Teloschistaceae is divided into 3 recognized subfamilies: Xanthorrhoideae, Coloplacoideae, and Teloschistoideae. In 2015, researchers proposed a 4th subfamily, Brownleealloideae, which was later shown by genetic studies to be a grouping based on mixed or misinterpreted data rather than a distinct lineage. Further analysis placed what was thought to be Brownleealloideae within the already established Teloschistoideae, suggesting the proposed subfamily was not a separate branch of the family tree. DNA evidence also dispersed members of the informally introduced subfamily Icrioideae across the 3 acknowledged subfamilies, primarily within Teloschistoideae. Despite this, Sergei Kondratyuk and colleagues continue to use Brownleealloideae and Icrioideae in their publications, assigning 9 genera to the former and 2 to the latter. The well-supported subfamilies, Xanthorrhoideae, Coloplacoideae, and Teloschistoideae, encompass a range of growth forms, crustose, foliose, and fruticose, demonstrating the diverse evolutionary paths within the family. These groups are genetically distinct, each subfamily showing unique patterns in their nuclear large ribosomal subunit RNA sequences. Coloplacoideae Arup, Sting and Froden, 2020. Type genus, Coloplaca. Proposed by Esther Gaia and colleagues in 2012 and validly published in 2020, Coloplacoideae consists mostly of crustose lichens with a wide geographical spread and produces a range of unique chemical compounds. Teloschistoideae Arup, Sting and Froden, 2020. Type genus, Teloschists. Initially proposed in 2013 and validly published with a full diagnosis in 2020, this subfamily is predominantly found in the southern hemisphere. Xanthorrhoideae Arup, Sting and Froden, 2020. Type genus, Xanthoria. Named by Gaia and colleagues in 2012 and formally validated in 2020, Xanthorrhoideae species are primarily distributed in the northern hemisphere. The order Teloschistaceae was first proposed by David Hawksworth and Erickson in 1986, with a single family, Teloschistaceae. Other families were added later. In the 1990s, several authors recognized the Teloschistaceae as a suborder within the Lecanoorales. As a suborder it was named Teloschistaceae. Following the appearance of preliminary molecular studies, the Teloschistaceae was classified by some within the order Lecanoorales, although others maintained the Teloschistaceae as a valid order. A large-scale, multi-gene phylogenetic study of the class Lecanoormycetes published in 2014 corroborated the ordinal status of the Teloschistaceae, and showed it comprises 2 clades, Letroiridinea, containing Brigantiaceae and Letroirichaceae, and its sister clade, Teloschistaceae, containing Teloschistaceae and Megalospiraceae. The suborder Teloschistaceae was formally proposed by Esther Gaia and François Lutzany in 2016. Historically, classification of taxa within the family relied on physical characteristics such as growth form, the nature of the outer layer of the lichen, the cortex, and spore type. Studies using modern molecular phylogenetics have shown that phenotypic characteristics, characters, are not always reliable markers of phylogenetic relationships, and classifying species based on these characters has occasionally led to inaccurate interpretations of their evolutionary history. Advanced DNA analysis techniques have allowed scientists to identify and differentiate cryptic species, which, though visually indistinguishable, are genetically distinct. This approach has unveiled distinct species within previously thought homogeneous groups, like the genus Coloplaca, by uncovering their unique genetic markers. Although Teloschistaceae is now well represented in GenBank, with thousands of DNA sequences, the early molecular studies were limited by having too few examples of each species to draw definitive conclusions. With the increasing availability of genetic sequences, researchers began to gain a better understanding of the family's phylogeny. One significant finding from molecular data is that the traditional morphological methods had mistakenly grouped different species together. For example, The genus Coloplaca was once thought to be descended from a single lineage, i.e. monophyletic, but is now understood to have been composed of multiple, unrelated groups, polyphyletic. This insight has prompted numerous proposals to redefine the genus into smaller, monophyletic groups, but such taxonomic changes have sometimes met with resistance due to the vast number of species reclassifications they would entail. According to the lichenologist Robert Lucking, families like Teloschistaceae, which have undergone several changes in genus classification through various studies, require phylogenetic consolidation through extensive multi-locus analysis, incorporating all available data and employing rigorous analytical methods. This strategy, akin to approaches taken with families such as Collemataceae, Graphidaceae, Panariaceae, and Parmeliaceae, is essential for accurately revising the taxonomic classification of this diverse and widespread group of lichens. Molecular evidence has also helped to map the family's relationships within the class Lecanorhamycetes. A 2018 study identified the Megalospiraceae as the Teloschistaceae's closest relative. In general, Teloschistaceae members are known for their vibrant colors, spanning a spectrum of yellow, orange, and red hues, attributed to anthraquinone pigments. This group of lichens has a broad range of physical forms, from thin, encrusting, crustose, to leaf-like, foliose, or even bushy, fruticose, formations. Although it is an atypical growth form for the Teloschistaceae, members of genus Ioplaca are somewhat umbilicate, meaning they have a somewhat circular, leafy thallus attached to the substrate at a single point. In contrast to its barely visible thallus, the apophysia of Athalia holocarpa are prominent. Like most Teloschistaceae, these apophysia are lecanorrhine in form, rounded with thick, pale, thalline margins. Teloschistaceae lichens have a symbiotic relationship with a photobiont, generally a member of the green algal genus Treboxia. The lichen's reproductive structures, or ascomata, are usually brightly colored, and typically in the form of an apophysium, a wide, open, saucer-shaped, or cup-shaped fruit body. In most species, these apophysiate ascomata have a lecanorrhine form, in which the apophyseal disc is surrounded by a pale rim of tissue known as a thalline margin. Fewer Teloschistaceae species have biaterine or lessidane forms, in which the apophyseal disc lacks a thalline margin. Reproductive propagules, such as Isidia and Sordia, can be found in select species. The ascomata encase asci, cylindrical formations that commonly contain between 4 and 16 ascospores, with 8 being the most prevalent count. These asci are characterized by a well-developed J and layer amyloid cap. The term J+ refers to the positive staining reaction of the ascus tip to iodine, specifically when it turns blue or dark blue in the presence of iodine-based solutions like Melzer's reagent or Lugol's iodine. The internal apical structure of the ascus is rudimentary, relative to the more complex apical structures in other related families. Teloschistaceae ascospores, typically translucent, usually have 1 to 3 septa, internal partitions, with a prominent central septum connected by a canal to the spore's internal cavities, or lumina. Although the presence of a two-chambered, polarilocular, structure in these ascospores is generally indicative of the Teloschistaceae, the spores lack other distinctive features that could be useful as defining taxonomic characteristics. Historically, polarilocular ascospores were regarded as a hallmark of the Teloschistaceae. The incorporation of genera such as Apatoplaca, Cephalophysis, Fulgensia, and Xanthopeltis, which have either non-septate or simply septate spores, has necessitated a reassessment of what fundamentally characterizes this group. A distinctive feature of Teloschistaceae is the presence of the gelatinous paraphyses, filament-like support structures in the reproductive apparatus, with either unbranched or slightly branched structures culminating in bulbous ends. Within this family, asexual reproduction leads to the creation of pycnidia-type conidiomata, i.e., small, flask-shaped fruiting bodies, producing translucent asexual spores, conidia, that are either bacillar, rod-shaped, or bifusiform, double-spindle-shaped. The tissue composition of the thallus and apophysia is characterized by a loosely paraplectend chimatous structure, meaning the constituent fungal hyphae are oriented in different directions. Regarding morphological variety of Teloschistaceae, cruise toes, leprose, Leproplaca chrysotida. Fruticulose, Polycoleonocoralloides. Cruise toes, Placodioid, Variospora aurantia. Lichenicolous lichen Irichanzenia epithalina, red, growing on the cruise toes Rhizoplaca novomexicana. Foliose, Xanthomendoza oregana. Foliose, Xanthoria parietina. Squamulose, Squamulia squamosa. In lichens, photobionts are the photosynthetic organisms that collaborate with fungal partners to enable the unique lichen symbiosis. Members of the Teloschistaceae associate with Treboxioid green algal photobionts, i.e. resembling or belonging to the green algal genus Treboxia. An early study investigating the ultrastructure of the interaction between the fungus and alga in several Teloschistaceae species showed that, in most cases, the cells were merely in close proximity to one another, with only a few instances of fungal cells invading the algal cells. The widespread Xanthoria parietina species complex has been identified to associate with various Treboxioid species, including Asterochlorus italiana, Treboxia arboricola, and T. decolorans. Within the order Teloschistaceae, unlike the Teloschistaceae, species in the families Letroerichaceae and Megalospiraceae primarily partner with the green algal genus Dictyochlorops. Due to their resilience to desiccation, Treboxia species serve as the main photobionts for lichen-forming fungi found in extreme environments such as the Antarctic, Arctic, alpine regions, and deserts, where lichens face continual exposure to intense dryness and temperature shifts. Studies of photobionts in the Teloschistaceae, including Foliose genera, Xanthoria, Xanthomendoza, and a fruticose genus, Teloschists, demonstrate a consistent association with specific Treboxia clades. This finding suggests a genus-level specificity, with only select Treboxia subclades forming symbiotic relationships. This specificity is not absolute and may vary with habitat. Lichens in extreme climates have been observed to associate with a broader range of photobionts. Parietin, top, and the structurally similar emodin, bottom, are anthraquinone pigments common in the Teloschistaceae. The predominant chemical compounds found in the Teloschistaceae are pigments known as anthraquinones. These substances, which are deposited in the upper cortical layer of the lichen, have photoprotective properties, as they can absorb ultraviolet, UV, and blue light. The development of secondary metabolites, particularly anthraquinones, has been essential in the evolutionary adaptation of the family to diverse environments, enabling their transition from shaded, plant-based habitats to sun-exposed, arid regions and contributing significantly to the success of the familial lineage. A 2023 study used comparative genomics to identify a metabolic gene cluster involved in anthraquinone metabolism and shared uniquely across the Teloschistaceae. Phylogenetic analyses of fungal polyketide synthases, PKSs, reveal a consistent grouping of genes, hinting at a shared ancestral trait for anthraquinone biosynthesis within the subphylum Pezizomycotina. Between 1897 and 1906, the mycologist Friedrich Wilhelm Zopf and the chemist Oswald Hesse conducted a series of early chemical studies on members of the Teloschistaceae, leading to the extraction of the reddish pigment parietin from selected species. Parietin is an antioxidant molecule that is produced in greater amounts in lichen thalli that are exposed to excess nitrogen. In a 1970 publication, Johann Sanderson surveyed 230 Coloplaca species for anthraquinones as part of a phytochemical study of the Teloschistaceae, and concluded that the studied species could be arranged according to their anthraquinone content in 13 chemical groups. In 1997, Ulrichting analyzed secondary metabolites from species of Coloplaca, Teloschistaceae, and Xanthoria to look for chemical patterns of consistent combinations and proportions of lichen products. He identified two chemosyndromes, characteristic sets of chemical compounds, with periodin, amidin, folicinol, folicinol, and periodinic acid as the main substances. Periodin acts as a UV light filter to provide optimal light intensities for the photobionts that are resident in the internal algal layer. Studies show that in Xanthoria periotina, the more light the lichen is exposed to, the higher the concentration of periodin. In the Teloschistaceae, periodin may also serve a defensive role. In the Negev Desert, the periodin-containing Teloschistaceae species Elenchiniana ehrenbergii and Cerophorolacunosata are avoided by grazing snails, but they frequently consume lichens like Diploaceae canescens and Bulia subulbula, both in family Colichaceae, which lack periodin. In their large-scale phylogenetic analysis of the Teloschistaceae, the lichenologists Ulf Arub, Ulrichting, and Patrick Froden analyzed about 4,000 specimens of members of the family using high-performance liquid chromatography, and identified more than 100 secondary metabolites, mostly anthraquinones. They noted that in a large majority of cases, the distribution of lichen products was more or less constant within species. In some instances, the secondary chemistry is important at higher taxonomic levels, i.e., ranks higher than species. For example, the genus Catenaria is characterized by the presence of 7-chlorocatanarin, a secondary metabolite previously unknown in lichens. The substance usnic acid characterizes the genus Usnichroma, and 5-chloroimidin occurs in all but one species of Shackletonea. The secondary chemistry of the Coloplacoideae is the most diverse amongst the three Teloschistaceae subfamilies, as it contains both chlorinated anthraquinones and depsidins. Apothecia of Icrea Ceroceaceae grown in full light develop completely black margins resulting from the accumulation of the pigment cineroorphagrine. Although most Teloschistaceae lichens produce anthraquinone pigments in shades ranging from yellow to orange to red, the genera Apatoplaca and Cephalophysis lack these anthraquinones. Similarly, the genus Pyrenodesmia encompasses species where anthraquinones are absent and replaced by substances such as cineroorphagrine or Sedifolia gray. These insoluble lichen pigments may confer UV protective abilities similar to anthraquinones. Taxa of the closely related genera Kettlingeria and Sanguiniodiscus have anthraquinones in their apothecia and Sedifolia gray in their thalliae. The species Kettlingeria neotorica features apothecia of two color variants, orange-red, with anthraquinones, and gray, with Sedifolia gray. The absence of anthraquinones is not a synapomorphic character, but appears independently in unrelated lineages of Teloschistaceae. As such, it is a phylogenetically unreliable character. Adaptive radiation in the Teloschistaceae has been studied to understand the key phenotypic changes leading to their diversification. This diversification is believed to be connected to the spread of anthraquinone pigments in their thallus. Initially, these pigments were thought to have appeared during the Teloschistaceae's first divergence, with a more widespread occurrence developing later. The distribution of anthraquinones in Teloschistaceae lichens varies, from being dispersed across the organism's surface to localized regions. Analysis suggests that the family's lineage witnessed a loss and subsequent return of these pigments over time, considering their presence in the thallus and apothecia as the ancestral state. Ecologically, these organisms transition from inhabiting shaded bark environments to colonizing sunlit, rocky areas during their diversification. The analysis of phenotypic traits and diversification rates shows that anthraquinones in the thallus and greater sun exposure have contributed to an acceleration of diversification. On the contrary, living in shaded environments or having a crustose continuous, smooth, non-scaly, growth form hinder diversification. The choice of substrate, be it rock or bark, did not have a pronounced impact on diversification rates. This adaptive radiation within the Teloschistaceae is estimated to have initiated around 100 million years ago, specifically during the late Cretaceous period. Factors like climatic shifts, continental separations, and the emergence of flowering plants are theorized to have influenced the adaptive landscape. Such factors might have promoted the development of light-protective anthraquinones, enabling Teloschistaceae to colonize exposed environments. The classification and number of species within the Teloschistaceae have evolved significantly over time. Historical estimates have varied, with 10 genera and 47 species recognized in 2001, increasing to 12 genera and 644 species by 2008. The new classification proposed by Arup and colleagues in their 2013 taxonomy article recognized 39 genera, including 31 that were newly described or resurrected. By 2016, there were between 51 and 53 genera and about 700 species, 65 genera and 755 species in 2017, and 71 genera and about 840 species by 2022. In the same year, Kondratiuk and colleagues enumerated all members of the Teloschistaceae with publicly available DNA sequences, and confirmed 590 species across 115 genera. As of January 2024, Species Fungorum, in the Catalogue of Life, accepts 117 genera and 805 species in the Teloschistaceae. The largest genus is Coloplaca, at 173 accepted species. In terms of species diversity, Teloschistaceae stood as the sixth largest lichen-forming fungal family by 2017, following Parmeliaceae, Graphidaceae, Verrucaraceae, Ramalinaceae, and Lecanoraceae. Each genus is paired with its taxonomic authority, denoting the first describers using standardist author abbreviations, the publication year, and the number of species. Genera are organized by subfamily. Examples from subfamily Coloplacoideae, clockwise from upper left Blasnia ferruginea, Coloplaca maculata, Ignioplaca igna, and Kettlingeria erythrocarpa. Apatoplaca Pold and Hafelner, 1980, 1 sp Blasnia A. Massel. 1852, 11 species Bryoplacasting, Froden and Arup, 2013, 3 species Coloplaca th Father, 1860, 351 species Cephalophysis, Hurdle, H. Chilias, 1985, 1 sp Ilifdalia s.y. Condra, Carnifold, Ilix, A. Thel and Her, 2014, 2 species Elenchiniana s.y. Condra, Carnifold, Ilix, A. Thel and Her, 2014, 3 species Freya s.y. Condra, LKS and Her, 2016, 7 species Franwilcia s.y. Condra, Carnifold, Ilix, A. Thel and Her, 2014, 3 species Fulgensia A. Massel. And not. 1853, 2 species Ginteracela s.y. Condra. And Her, 2017, 1 sp Galalechia A. Massel. 1852, 40 species Hanchtrascia s.y. Condra. 2017, 2 species Hunechia s.y. Condra, Ilix, Carnifold, A. Thel and Her, 2014, 4 species Ioplaca Pold, 1977, 2 species Jacinhuria s.y. Condra, LKS and S.O.O, 2015, 1 sp Clotteruela s.y. Condra. And Her, 2017, 3 spp Kettlingeria trevis. 1857, 15 species Lacrima bungarts, Arupansting, 2020, 4 species Londonia s.y. Condra, LKS and Her, 2017, 2 species Lendi Mariella s.y. Condra. 2020, 9 species Leproplaca, NYL. 1888. 7 species Loicoecia S.Y. Condra, S.O. and her. 2015. 3 species Marchantiana S.Y. Condra, Carnifold, Ilix, A. Thel and her. 2014. 7 species Micomia S.Y. Condra, Carnifold, Ilix, A. Thel and her. 2014. 4 species Neoplaca I.V. Frolov, Prokopiev and Konoriva. 2023. 1 sp obscura Placasting, Arup and Bungards. 2021. 3 species Oceanoplaca Arup, Sting and Bungards. 2020. 6 species Node 6 Oleg Blumia S.Y. Condra, LKS and her. 2020. 1 sp Opalsha S.Y. Condra and LKS. 2017. 4 species Oxnarigopsis S.Y. Condra, Upreti and her. 2017. 4 species Piscioshaella S.Y. Condra, LKS and Farkas. 2020. 6 species Pyrenodsmia A. Massel. 1852. 6 species Rufoplaca Arup, Sting and Froden. 2013. 10 species Sanguiniodiscus I.V. Frolov and von Drack. 2020. 4 species Cirrhophora Pold. 1983. 8 spp. Socioplaca Bungards, Sting and Arup. 2020. 1 sp Aprecia S.Y. Condra, A. Thel and her. 2017. 2 species Usnachromosting, Arup and Froden. 2013. 2 species Variospora Arup, Sting and Froden. 2013. 16 species Xanthoptychia S.Y. Condra and Revera. 2017. 3 species Yoshimuria S.Y. Condra, Carnifold, Ilix, A. Thel and her. 2014. 4 spp. Examples from subfamily Teloschistoideae, clockwise from upper left Nia Brownleella cinnabarna, Neorma Haciaceana, Wet Mariana broadii, and Fulgigasparia apprsa. Eretoplaca Wilk, Pavigan and Lucking, 2021. 1 sp Brownleella S.Y. Condra, Carnifold, Ilix, A. Thel and her. 2013. 2 species Catenarinasting, Zgiard, Arup, Elvbacandilix. 2014. 3 species Cinnabaria Wilk, Pavigan and Lucking. 2021. 1 sp Elixjonia S.Y. Condra and her. 2017. 4 species Filsoniana S.Y. Condra, Carnifold, Ilix, A. Thel and her. 2013. 9 species Falmania C.W. Dodge, 1967. 2 species Fulgigasparia S.Y. Condra, M.H. Zhang, Carnifold, Ilix, A. Thel and her. 2013. 5 spp Haloplaca Arup, Sting and Froden. 2013. 3 species Harrisovskia S.Y. Condra. 2017. 1 sp Haciaceae S.Y. Condra, L.LKS, Carnifold and A. Thel. 2018. 3 species Icrea S.Y. Condra, D. Upreti and her. 2017. 2 species Iqbalia phayas, Afshan and S.Y. Condra. 2022. 1 sp Joseph Polchia S.Y. Condra and Carnifold. 1997. 3 species Carnifia S.Y. Condra, Ilix, A. Thel and her. 2013. 3 species Lazarenkoiopsis S.Y. Condra, LKS and her. 2017. 1 sp Loicois Laszlo S.Y. Condra, Carnifold, A. Thel and her. 2019. 2 spp Nia Brownleella S.Y. Condra, Ilix, Carnifold and A. Thel. 2015. 5 species Nevelia S.Y. Condra and her. 2017. 2 species Neorma A. Massel. 1861. 5 species Resaniana S.Y. Condra, Carnifold, A. Thel, Ilix and her. 2015. 1 sp Remanula S.Y. Condra and her. 2018. 5 species Scuteriasting, Arupanfroden, 2013, 1 sp Sirenophilisting, Arupanfroden, 2013, 4 species Stellarangifroden, Arupansting, 2013, 3 species Stramoniella s.y. Condra, Carnifold, A. Thel, Ilix and her, 2015, 4 species Terrasginia s.y. Condra, Carnifold, A. Thel, Ilix and her, 2015, 2 species Tassilo s.y. Condra, Carnifold, A. Thel, Ilix and her, 2015, 2 species Tailriolina s.y. Condra, Carnifold, A. Thel, Ilix and her, 2016, 2 species Teloschistaceae Norman, 1852, ca 24. Species Teloschistaceae Froden, Stingandarup, 2013, 3 species Theliana s.y. Condra, Carnifold, Ilix and her, 2015, 1 sp Villaforesting, Arupanfroden, 2013, 9 species Wetmariana Arup, Stinganfroden, 2013, 2 species Wiltalia s.y. Condra, 2021, 1 sp. Examples from subfamily Xanthorrhoideae, clockwise from upper left Athalia scopularis, Cologaea saxicola, Diaphorea ligulata, and Xanthocarpia crenulatella. Amenceniasting, Garadoban, Arupanfroden, 2014, 2 species Athalia arup, Frodenansting, 2013, 17 species Austroplacasting, Frodenandarup, 2013, 10 species Cologaea arup, Frodenansting, 2013, 19 species Cirathalia arup, Frodenansting, 2013, 4 species Charcoianasting, Garadoban. Andarup, 2014, 1 sp Copinziella s.y. Condra. And LKS, 2018, 3 species Digigilla s.y. Condra. And L.LKS, 2017, 2 species Diaphorea ach. 1809, 25 species Irichanzenia s.y. Condra, Carnifold and A. Thel, 2020, 3 species Flavaplaca arup, Stinganfroden, 2013, 28 species Fominiella s.y. Condra, Upreti and her, 2017, 2 species Gallowayella s.y. Condra, Fedor & Co., S. Stenrus, Carnifold, Ilix and A. Thel, 2012, 15 species Gallikovia s.y. Condra, Carnifold, Ilix, A. Thel and her, 2014, 1 sp Gondwaniasting, Frodenandarup, 2013, 4 spp Hanegaria s.y. Condra, Fedor & Co., S. Stenrus, Carnifold, Ilix, her and A. Thel, 2012, 1 sp Uriella s.y. Condra. 2017, 5 species Ignioplaca s.y. Condra, Carnifold, Ilix, A. Thel and her, 2014, 1 sp Jackalixia s.y. Condra, Fedor & Co., S. Stenrus, Carnifold and A. Thel, 2009, Jesmerea s.y. Condra, Fedor & Co., S. Stenrus, Carnifold, Ilix, her and A. Thel, 2012, 1 sp Kudratavila s.y. Condra, L.LKS, Carnifold and A. Thel, 2022, 5 species Langiatia s.y. Condra, Carnifold, Ilix, A. Thel and her, 2014, 2 species Lazarencoella s.y. Condra, Carnifold, A. Thel, Ilix and her, 2015, 2 species Martinionzia s.y. Condra, Fedor & Co. S. Stenrus, Carnifold, Ilix, her and A. Thel, 2012, 1 sp Masjukila s.y. Condra, Fedor & Co., S. Stenrus, Carnifold, Ilix, her and A. Thel, 2012, 8 species Orientophila arup, Stinganfroden, 2013, 15 species Ovialbornia s.y. Condra, Fedor & Co., S. Stenrus, Carnifold, Ilex and A. Thel, 2009. 3 species Oxneria S.Y. Contra. And Carnifold, 2003. 4 spp. Packetalisting, Arup and Froden, 2013. 7 species Parviplaca Arup, Sting and Froden, 2013. 6 species Polycolia Nahu, 1908. 18 species Rusifskia S.Y. Contra. And Carnifold, 2003. 19 species Scythioria S.Y. Contra. Carnifold, Ilex, A. Thel and Her, 2014. 3 species Seawardiella S.Y. Contra. I. Carnifold and A. Thel, 2018. 2 species Shackletoniasting, Froden and Arup, 2013. 5 species Solitaria Arup, Sting and Froden, 2013. 1 sp Squamulia Arup, Sting and Froden, 2013. 15 species Tuvodiana S.Y. Contra. And Her, 2017. 3 species Tomnashia S.Y. Contra. And Her, 2017. 4 species Transdrachia Sting and Arup, 2023. 2 species Varicoplaca S.Y. Contra. Carnifold, Ilex, A. Thel and Her, 2014. 1 sp Xanthocarpia A. Massel. And the Knot. 1853. 12 species Xanthodactylon P. A. Yuvan. 1941. 2 species Xanthocataroa S.Y. Contra. Fedor & Co. S. Stenrus, Carnifold, Ilex and A. Thel, 2009. 2 species Xanthomendoza S.Y. Contra. And Carnifold, 1997. 20 species Xanthopeltis R. Sant. 1949. 1 sp. Xanthoria, Father, Th. Father, 1860. 10 species Xeravila S.Y. Contra. And Her, 2015. 8 spp. Some of the genera proposed during the restructuring of the family have since been shown to be nomenclaturally illegitimate or unavailable for use. For example, Andina Wilk, Pavigen and Lucking, 2021, has been replaced with Wiltalia. Pheoplacasting, Arup and Bungards, 2020, has been replaced with Obscuraplaca. Tailrila S.Y. Contra, Carnifold, A. Thel, Ilex and Her, 2015, has been replaced with Tailrialina. The scrambled egg lichen, Gyalelechia fulgens, is a ground-dwelling species and a component of some biological soil crusts. This rock in Gaspereau Lake is frequented by great blackback gulls, creating the conditions for a localized nitrogen-rich environment conducive to the growth of the orange lichen Rusifskia elegans. The family has a worldwide distribution, although members occur predominantly in temperate regions, and in semi-arid regions of the subtropics. Most members either grow on rock, Saxicolous, or on bark, Corticolous. As an exception to this general ecological preference, the genus Bryoplaca contains species that only grow on mosses and detritus. Some Fulgensia species grow on soils, particularly those rich in lime. Several Crustose Teloschistaceae species, typically Saxicolous in nature, have been recorded growing on human bone remains recovered at a looted late Holocene aboriginal cairn burial site in Argentina. Many species of the family are moderately to strongly nitrophilous, meaning that they prefer habitats rich in nitrogen, particularly in the form of nitrate. Adapted to environments with high sunlight exposure, Teloschistaceae lichens show an enhanced ability to fix carbon from the atmosphere, a crucial process for their sustenance and growth. These lichens also have an increased capacity to absorb excess nitrogen, a trait linked to their adaptation to intense light conditions. This dual adaptation, improved carbon fixation and nitrogen absorption, is evident in the family's small folios and crustose species, which show greater tolerance to elevated nitrogen levels. The absorption of additional nitrogen is thought to be associated with the presence of specific compounds like anthraquinones in these lichens. These compounds not only contribute to their characteristic yellow/orange coloration but may also help protect fungal cell membranes. Xanthoria parietina is one example of a widespread lichen that appears to be experiencing an increase in its range due to its ability to tolerate nitrogenous pollutants, and its potential ability to displace native lichen species as a result. Coloplaca, Fulgensia, Teloschistaceae, and Xanthoria are genera that are characteristic of sun-exposed habitats. In some extreme desert environments, Coloplaca, in the broad sense, including lichens historically classified in this genus, may be the only genus present. Coloplaca and Xanthoria dominate harsh coastal environments. There are several Teloschistaceae genera that contain lichen-dwelling, lichen-dwelling, species. These non-lichenous fungi originate from subfamily Coloplacoideae, Coloplaca, 26 species, Gyalelechia, 1 sp, Variospora, 1 sp, from subfamily Teloschistoideae, Catenorina, 1 sp, Cyrenophylla 1, and from subfamily Xanthorioideae, Flavoplaca, 4 species, Pachydeltus, 1 sp, and Shackletonea, 3 species. Lichen species within the Teloschistaceae generally have a broad range of hosts. Their geographical distribution seems to be influenced not just by the classification of their host lichen, but also by the substrate they grow on. Teloschistaceae has a high diversity in polar regions and a substantial number of bipolar species, i.e., species occurring in both northern and southern hemispheres but largely absent from intermediate, tropical latitudes. Examples include Gallowayella borealis, Austroplaca sauropelta, and Scythoria flogina. There is a relatively low diversity of cruise-toes Teloschistaceae in Central Europe. Localized exceptions occur primarily in sunlit locations with either calcareous or nutrient-rich siliceous rock formations. These habitats are predominant in the alpine regions of the Alps and the Carpathian Mountains, as well as in the arid, warm rocky steppes. Some Teloschistaceae genera have a strong geographic center of species richness. Examples include Elixjonia, Australasia, Orientophylla, East Asia, Shackletonea, Antarctic and Subantarctic, Stellarangea, Southwestern Africa, and Xanthoria, Mediterranean area. Several studies published since 2014 have enumerated the Teloschistaceae taxa occurring in certain defined geographical areas. These include Altai-Sayan region, 103 species and 31 genera. Crimea, 119 species. Dagestan, 85 species. India, 115 species and 36 genera. Italy, about 160 species. Galapagos Islands, 24 species. Mexico, 142 species and 6 genera. New Zealand, about 100 species. Russian Far East, 84 species. Ural, 81 species. The Lichenicalous Tremelicoloplaceae species complex on Teloschistaceae hosts, A. on Xanthoria parietina, B. on Rusifskia elegans, C. on Variospora flavescens, D. on Rusifskia sordida, E. on Cologaia piscilla, F. on Xanthocarpia sp. White circles enclose tremale-induced galls. Scale bars equals 1 mm. Teloschistaceae species host a variety of lichenicalous fungi. Some, such as Cercidospora caudata and Stigminium cyrenee, infect a wide range of hosts within the family. More generally, these parasitic fungi have a preference for specific species or genera of Teloschistaceae. An example is the relationship between Teloschistaceae lichens and the fungus Tremelicoloplaceae. Integrative studies combining molecular data and ecological approaches revealed at least 6 distinct lineages of T. coloplaceae, each specialized to a particular host, indicating a complex of closely related species. This diversification of T. coloplaceae appears to have occurred in tandem with the rapid diversification of the Teloschistaceae since the late Cretaceous period, implying coevolution. Further molecular studies have delineated the T. coloplaceae group into a complex of at least 9 distinct species. Out of these, 5 new species were formally described in 2023, each adapted to a single host species or genus within the Teloschistaceae. The Cruz-Toes ignioplaca igna covers walls of the Fortaleza de Santa Teresa, a 260-year-old military fortification in Uruguay. Although Teloschistaceae species are not economically significant, their tendency to grow on rock surfaces has been documented to cause damage to marble structures. In some cases, the lichens, the major contributor of which was Xanthocarpia ferocissima, penetrated up to 10 mm, 3/8 inches, into the stone along larger cracks and 0.05 mm, 1500 inches, beneath loose surface crystals, leading to crumbling of the marble surface. Coloplacus pseudopolyatera and C. cuculifer are two Cruz-Toes species responsible for the slow degradation of the Konark Sun Temple in India. In regional practices, Teloschistaceae elegans is used for treating wounds. In Afghanistan, it is applied directly. In Kyrgyzstan, the lichen is mixed with butter and used as a remedy for diarrhea in livestock. Teloschistaceae flavicans is used in China for its purported properties of clearing heat in the lung and liver, and removing toxins. Oxynuria fallax has been incorporated into traditional Tibetan medicinal treatments. The presence of Xanthoria parietina in urban ecosystems, even in high-pollution areas, offers potential insights for biomonitoring. Although its tolerance to air pollution allows it to persist where other species may not, its presence can be used in conjunction with more pollution-sensitive species to gauge overall air quality. In polluted environments, the health and abundance of X. parietina, relative to other less tolerant species, can provide data on the level and impact of urban pollution. Teloschistaceae elegans has been studied in experiments where specimens were exposed to outer space conditions, including extreme temperatures, ultraviolet radiation, and ultra-high vacuum. The results demonstrated the lichen's substantial capability to endure these conditions. Coloplacca alanii is a poorly known New Zealand endemic. The conservation status of 3 Teloschistaceae species has been assessed for the global IUCN Red List. Coloplacca rhinodonilb, vulnerable, 2017, is at risk from tourism development and increased erosion on Sardinia's coasts. Xanthoptychia arantaica, endangered, 2020, faces multiple threats due to climate change in the Canadian Arctic. These include habitat loss from rapidly eroding coasts, increased sea ice melt, saline washed from storm surges, and permafrost melting. Further, the changing climate may allow the advancement of southern vegetation communities and the introduction of invasive species, potentially exacerbating the impact on this lichen by altering its native habitat. Teloschistaceae bruensis, critically endangered, 2021, is at risk due to multiple threats in Peru and Chile, including potential development, habitat fragmentation, for X4 races like the Dakar Rally, air pollution, and the impact of livestock like goats and cows, which alter the habitat through grazing and trampling. Other Teloschistaceae members, some with limited geographic distributions, make appearances on regional red lists. For example, the Cruz-Toes New Zealand endemic Coloplacca alanii, first documented in 1932, was not collected again until 81 years later. Because of its sparsity and small total area of occupancy, it has been assessed as threatened/nationally critical in the New Zealand threat classification system. In some large geographical areas, the full extent of the diversity of Teloschistaceae taxa is not well known. Examples include South America, where the family has not historically received much attention, and China, where of 2,164 lichen species evaluated for inclusion on its red list, only 49 were members of the Teloschistaceae, 13 of those were listed as least concerned species, and the other 36 as data deficient.